Hi friends, welcome to my video. In this video, we are going to see about bacterial transformation. Transformation is the direct uptake, incorporation and expression of foreign DNA or genetic material from the environment. Bacterial transformation is a major mechanism of horizontal gene transfer. Let's see, what is horizontal gene transfer? Horizontal gene transfer is the non-sexual movement of genetic material between unicellular organisms or between unicellular and multicellular organisms or between multicellular organisms. Incoming genes can replace or introduce new genes into a genome. That is, horizontal gene transfer is a process in which an organism transfers genetic material to another organism that is not its offspring. Horizontal gene transfer, also known as lateral gene transfer. Horizontal gene transfer is an important factor in the evolution of many organisms. Bacteria have acquired a variety of important traits, including antibiotic resistance, pathogenesis, and metabolic pathways via horizontal gene transfer. Transformation has been studied mainly in pneumococci, bacillus species, and Haemophilus influenza. Pieces of DNA involved in transformation may carry about 10 to 50 genes. The process of gene transfer by transformation does not require a living donor cell, but only requires the presence of free DNA in the environment. Once the DNA enters the cytoplasm, it may be degraded by nucleases or may integrate into the chromosome. Transformation works best with DNA from closely related species. The naturally competent bacteria carry sets of genes that provide the protein machinery to bring DNA across the cell membrane. You should note one important point here, that is, only single-stranded DNA may pass through. One strand is therefore degraded by nucleases. The translocated single-stranded DNA may then be integrated into the bacterial chromosome. One more point to be noted here is that due to the differences in structure of the cell envelope between gram-positive and gram-negative bacteria, there are some differences in the mechanisms of DNA uptake in these cells. DNA as a nutrient source. DNA is an excellent source of the deoxyribonucleotides needed for replication of the cell's own genome. Environmental DNA is ubiquitous in most bacterial environments, especially biofilms. Competent cells take up preformed bases and nucleotides where possible and also use extracellular DNA as a nutrient source. Most competent bacteria that have been tested can take up DNA from any source. Let's see the sum up of bacterial transformation. DNA has both biochemical and informational properties. The effects of DNA uptake depend on the nutritional needs of the cell, the presence of DNA damage, the ability of incoming DNA to recombine with chromosomal DNA. That is, high similarity with the chromosomes also allow extracellular DNA to change the cell's genotype by homologous recombination, a process called natural transformation. Otherwise, it is degraded and its nucleotide subunits are recycled. I am concluding this video here and see you soon in my next video. Thank you, friends.